Hola, buenos días a todos. Bienvenidos a este elevator pitch número 13 de la SEBBM. Hoy nos hemos ido al norte otra vez. Tenemos a dos estupendas investigadoras, Rosa Barrio y Laura Bozal, que van a hacer de moderadoras en esta sesión de hoy. Ya sabéis, como siempre, tenemos una charla de 20 minutos y luego 4 pildoritas cortas de 5 minutos con luego un tiempo para preguntas. Y al final de la sesión, una mesa redonda en la que tendremos invitados extra. Hoy nos van a acompañar la investigadora Rosa Barrio, del Centro de Investigación Cooperativa en Biociencias de Bilbao, y os voy a hacer una pequeña presentación de Rosa. Rosa se licenció en Biología en el año 88 por la Universidad Autónoma de Madrid y se doctoró también en Biología en el año 93 en esta misma universidad. Luego realizó una estancia postdoctoral con un contrato Marie Curie en el IMBB de Heraclión, en Grecia, y después fue postdoctoral fellow en el EMBL de Heidelberg, en Alemania, durante 5 años. En 2001 se incorporó como postdoctoral fellow en el SEBBM Severo Ochoa, en Madrid, y de 2003 a 2008 disfrutó de un contrato Ramón y Cajal, y desde 2004 dirige su propio grupo de investigación en el CIC bioGUNE, en Bilbao. Como veis aquí, el grupo de la doctora Barrio está interesado en la regulación del desarrollo mediante modificaciones posttraduccionales por miembros de la familia de la ubiquitina. Su grupo, por ejemplo, descubrió que la proteína sumo desempeña un papel importante en la ingesta de colesterol, contribuyendo a la síntesis de hormonas esteroideas mediante la regulación de distintos receptores, trabajo que se publicó hace poco en Nature Communications. También está interesada en la familia de proteínas SAL, necesarias para numerosos procesos biológicos, y han descubierto que mutaciones en SAL1 y SAL4 causan enfermedades raras, como la de Townsend-Brooks, y su grupo descubrió que los cilios primarios están alterados en células de individuos afectados y estudian la homeostasis de proteínas implicada en estas enfermedades. Este trabajo también se ha publicado en Nature Communications. La doctora Barrio además ha dirigido y participado en numerosas redes nacionales e internacionales relacionadas con el estudio de modificaciones posttraduccionales. El trabajo de Rosa, como veis aquí, ha dado lugar a más de 80 publicaciones, incluidas en revistas tan potentes como Nature, Nature Communications y Nature Cell Biology. Así que, Rosa, muchísimas gracias por estar con nosotros hoy en este elevator pitch.
Muchas gracias a vosotras. ¿Empiezo ya la presentación o vas a presentar a Laura?
Voy a presentar a Laura primero.
Vale,
y
estupendo.
Y así ya nos conocemos todos y ya te dejo en sus manos.
Muy bien.
Bien, pues como os decía, tenemos una segunda invitada hoy, que es Laura Bozal, investigadora postdoctoral en el CIC bioGUNE. Se doctoró en 2019 en Biología Molecular y Biomedicina en la Universidad del País Vasco, tras llevar a cabo su investigación predoctoral en el laboratorio de la doctora Barrio. Y realizó también una estancia predoctoral en el Memorial Sloan Kettering Cancer en Nueva York. En 2019 comenzó su investigación postdoctoral en el laboratorio del doctor Arcaiz Carracedo, miembro de la SEBBM y reciente Premio Nacional de Investigación, por cierto, también en el CIC bioGUNE. Y recientemente, tanto es así que la tenemos conectada desde Boston a las 6 de la mañana, está realizando una estancia postdoctoral en el Instituto Koch en el MIT de Boston. En la actualidad, su investigación se centra en comprender los mecanismos que llevan a la metástasis del cáncer de próstata. Así que, Laura, muchísimas gracias por acompañarnos y más en esta hora tan intempestiva para ti.
Hola, buenos días o buenas tardes allí. Un placer estar aquí con todas vosotras en esta tercera edición del Elevator Pitch. Y bueno, sobre la introducción de Rosa, una excelente investigadora. Cuando quieras, Rosa, the floor is yours.
Muy bien, muchísimas gracias. Voy a compartir pantalla. Estoy bien, ¿verdad?
Perfecto.
Vale, muy bien. Pues muchísimas gracias por la invitación a Sara y a María y a todo el equipo que organizáis estos Elevator Pitch. La verdad que son un éxito y os doy la enhorabuena y las gracias por organizarlos. Bueno, pues ya sin más dilaciones nos metemos un poco en arena. Como han comentado en la presentación, mi laboratorio está interesado en las modificaciones posttraduccionales y la homeostasis de proteínas, que básicamente... okay, I will go to the English, sorry, I was confused. So, as I was saying, my lab is interested in protein homeostasis and which is an intrinsic property of the cell, and basically it consists in this equilibrium of the proteins from their synthesis and folding until they exert their function and even their degradation. And in the mean moment between the folding and the function and degradation, the proteins, most of them, they suffer this, or they have these post-translational modifications, which are of many different natures, but we are interested in the modifications by the ubiquitin-like family. And the ubiquitin-like family is a wide family of proteins. They are small proteins. The most famous one is the ubiquitin, that gives the name to the family, but there are others like Net8 or Sumo, etc., that they are being more and more studied. And in fact, all the members of this session, they are interested in one or another of those proteins, and they are studying it in their context of their own scientific interest. So we will hear about their work on different of these ubiquitin likes. So these different ubiquitin likes, they share this modus operandi. They work a little bit in the same way. As I said, they are small proteins, and they are able, through their C-terminal part, to have a covalent bond to specific substrates, and through the action of different enzymes, for instance, the E1, E2, and the E3 ligases, which are important for the regulation. And this modified protein, as I said, is able to do its own function, change localization, interactions, activation, or even go to degradation. It is interesting to notice that this process is a reversible one by the action of the ubiquitinases or DAPs, and then the substrate is again liberated and go back to the initial state. So this protein homeostasis, this protein regulation, is very important for most of the cell processes and also organismal processes, including animal development, that, as you know, is a very highly regulated process and is very much conserved from, for instance, in animals, from vertebrates to invertebrates. And being such a regulated process, we know that if things go wrong, they can have terrible consequences. For instance, in humans, if there are problems during the development, they might give rise to these rare diseases or developmental diseases. This is, for instance, the case of the Townes-Brocks syndrome that we are studying in the lab. This is a rare disease, an ultra rare disease, I would say, that is caused by mutations in SAL1, in the factor SAL1. And the phenotypes of the people with this syndrome are here. These are these polydactyly, ear malformations, also heart and genitourinary malformations, ear defects, and polycystic kidneys, among others. And well, we thought that maybe these different phenotypes, they were reminiscent of those caused by cilia anomalies, which at the time it was difficult to associate Townes-Brocks with cilia, but these phenotypes actually, they call our attention. And I'm going to talk about the work of Laura Bozal while she was a PhD student in my lab on this matter. So, as I said, these Townes-Brocks patients, we ask ourselves whether they had any cilia anomaly. And just a brief reminder of what a cilia is. These are these protruding antennae in the cells, which are very important for signaling, for instance, sonic hedgehog signaling and other signaling pathways. And it arises, the cilia, from this mother centriole. This process is also highly regulated in the cell and in the organism. And actually, there are these two proteins here, this CCP110 and SEB97, that they reside in the top of the centrioles, that they are avoiding the growth of the centriole, which actually at certain moments regulated during the cell cycle, those proteins get degraded and the cilia is allowed to grow. So we ask ourselves then, as I said, whether the Townes-Brocks patients, they had cilia anomalies, and the answer is yes. In fact, they do. And what Laura saw is that in TBS cells and cells derived from patients, the cilia length is longer than in the control cells. And also the number of ciliated cells was also higher than in control cells. We know that this has also consequences in the Sony Hedgehog regulation in those cells. And to understand this, we ask ourselves how this SAL protein or mutations in the SAL protein could promote these longer and more frequent cilia. And it was not obvious because these SAL proteins, SAL1 among them, are transcription factors. They are zinc finger transcription factors. These oval color ovals here, they indicate the zinc fingers, and they normally reside in the nucleus. So it might be that this effect on the cilia is very indirect through the regulation of gene expression. But it happens, it was known that most of the mutations that they give rise to the Townsbrock syndrome reside in this area of the protein. As a consequence, the protein, the protein that is formed in the patients is a truncated protein that is lacking most of the zinc fingers. And interestingly, it resides now in the nucleus and in the cytoplasm. So we thought that maybe this difference in the localization between the full length and the truncated protein might be indicating a different interaction of the different forms of the protein in this case. So how to study that? To understand whether these two proteins, they had different interactor family, we went to look at the biotin based subprotein of these forms of the SAL1. So we use BIR-A. This is an enzyme from E. coli that is able to put a biotin in a particular sequence that is called ABITAC or BioTAC. But some people, I should say that this sequence doesn't exist in the cell. And some people, they saw that by making certain mutations, this specificity was lost. And the BIR-A enzyme was able to put biotins in any lysine that was close by. So they developed this, what is called this proximity proteomics, which is based on this promiscuous biotinylation of BIR-A, and it's called BioID. So we decided then to fuse BIR-A, this promiscuous BIR-A, to the full-length cell or to the truncated form. And we expected to see biotinylation of proteins in proximity, but not proteins that they were not in close proximity of the protein. Because of the strong binding between streptavidin and the BIR-A, we were able to isolate the biotinylated proteins and identify them by mass spectrometry. And we were very happy to see that, in fact, the truncated form is able to interact with these two proteins, the CCP110 and SEBB97, that are the ones that they were stopping the formation of the cilia. So that took us to the idea that maybe the presence of this truncated form was somehow depleting these stoppers of the cilia growth in the mother centriole. So we look at the cells again of the control cells or the Townsbrock cells, and by marking the CCP110 here in green or a marker of the mother centriole here in magenta, we could see that in the control cells, most of the cells will have two spots of CCP110 corresponding to the two centrioles, but that in the Townsbrock cells, we could see often that the CCP110 was missing from the mother centriole, indicating that then this mother centriole was able to grow a cilia. That was interesting, and we continue looking at these different interactors between the full length and the truncated form, and we found this protein that was very prominent in the list as specific for the truncated form. This is LUSP1, which at the moment was a not very much studied protein. And we saw that this protein was actually localized in the actin cytoskeleton and also in the centrosome, giving the possibility that maybe was some connection through this protein between these two parts of the cell. And interestingly, by knocking out the LUSP1, we saw that removing LUSP1 from the cells actually also made longer cilia. Those cells were making longer cilia, and the ciliated cells were more a higher proportion than in the wild type, which coincides with what we were seeing in the Townsbroks. Furthermore, we were able to see that in the Townsbrook cells, the levels of LUSP1 are very much reduced, both in the cytoskeleton and also at the centrosomes. So it was like LUSP1 was unstable in the TBS cells. And we saw this also by Western blot. In fact, the levels of LUSP1, we corroborated that were lower in the TBS cells versus the wild type. And that took us to think that maybe the presence of this mutant cell, this N-terminal part of the protein, was increasing somehow LUSP1 ubiquitination to make it more unstable. And that's what it seems to happen. Here you see this is a ubiquitination assay that we perform in the lab. And you can see that when we look at the Townsbrook cells, the level of ubiquitinated LUSP1 is higher than when we look in the wild type. So taking all this data together, we came out to this model in which we think that in the wild type cells, LUSP1 has an equilibrium between ubiquitinated and non-ubiquitinated forms, which is depending on the action of these E3 ligases and deubiquitinases, and that will give a steady state level of LUSP1 in the cell. But in the presence of this truncated part of SAL1, somehow this equilibrium is broken, and we get a higher ubiquitination of LUSP1, which drives to a higher degradation of the protein, prompting and facilitating the formation of cilia in the cells. This is the idea, but then one answer takes you to another question, and here we thought, okay, we ask ourselves what will be these factors that they will be regulating LUSP1, and they can be modified through the action of the N-terminal part of the protein. And to answer this question, we went back to our BIR-A and BioID experiments. In this case, we use another form of BIR-A that is called TurboID, which is a kind of smaller version of BIR-A, but is faster, but it works in the same way. And we perform promiscuous biotinylation or proximity proteomics using this TurboID. When we fuse it to LUSP1, we saw that actually LUSP1 was interacting with a number of deubiquitinases and also a number of E3 ligases. We also saw that the mutated form of SAL, the truncated form, is also interacting with a number of DAPs, or the ubiquitinases, also a number of E3s, and that it has been described that some of them, they are actually specific of the centrosome or microtubules, like the ones that we write here. So to try to understand which ones of those players will have a function in the regulation of SAL and of LUSP1 and eventually in the Townsbrook syndrome, that was the task of Veronica Muratori, PhD student in the lab, and that is now followed by Claudia Guinea in the lab, that they analyze how LUSP1 levels vary in when we remove or we overexpress these DAPs or these E3s. So because of the time constrictions, I will make a long story very short. But what we have seen is that actually USP21 might be regulating the levels of LUSP1. You can see here by silencing USP21, we get reduced levels by immunofluorescence and also by Western blot. And the counterpart is taken by MindBone1, which in a background depleted for MindBone, when we add the MindBone wild type, we get a reduction in the endogenous levels of LUSP1, indicating a possible regulation of LUSP1 by MindBone, while this doesn't happen in the control or when we overexpress a catalytically inactive form of the enzyme. So it seems like we believe that, although this is still work in progress, that MindBone and USP21, they are somehow interacting in this regulation of LUSP1 at the centrosome or in the cytoskeleton for the regulation of LUSP1. One thing that was clear from our work is that, and this I think I can be corroborated by any member of this panel today, is that to study protein modifications by the ubiquitin likes is challenging. And it's challenging mainly for two reasons. One is that it's a process that is very dynamic. As I told you, this process of the modification is actually reversible. So it is constantly being removed by the ubiquitinases of the cell. And also another problem that we have when we study these modifications is this low abundance. That means that the proportion of the substrate that is normally modified is very low compared with the pool of the protein in the cell, but although it has important consequences in the function. So the question was, how to study, for instance, the effect that a modification has on the interactors of a particular substrate. Is a protein, when it's not modified, interacting with the same factors than when the protein is modified by one of the ubiquitin likes? And to answer to this question, we needed to develop strategies to be able to see that in a more precise and specific way. And as you might have guessed by now, we went back to our favorite proximity proteomics strategy. In this case, again, with the turbo ID, and in this case, we combine this proximity proteomics with protein fragment complementation. And what we did is to cut this turbo ID in two halves, and none of them actually was functional. It could not work by itself. So those two parts, they were inactive. And the idea was as follows. We fuse one part, in this case the C-terminal, to one of the UBLs, in this case SUMO, and the other part, in this case the N-terminal of the turbo ID, to our substrate. So only when the substrate is SUMO-related, in this case, these two parts are close enough to reconstitute the active form of VRA. And only in that case we'll be able to biotinylate the proteins that they are in close proximity. So that means that we are able to mark only those substrates that they are interacting, only those proteins that they are interacting with the substrate when it is modified, and then identify them by streptavidin purification and mass spectrometry. So that was a nice idea on the paper. It was developed by Ori Barroso during his PhD in the lab, and by Jim Sutherland, who was the brain behind, one of the brains, together with Ori, behind the development of the whole strategy. And as I said, it will look nice on paper, but to see whether I was actually working, we took this proof of principle using the promyelocytic leukemia protein, or PML, that was fused, that is located in these nice nuclear bodies in the cell, and is a protein that is known to be SUMO-related and interact with SUMO. So we put SUMO fused to the one part of turbo ID and PML fused to the other part of turbo ID in the cells. And we were happy to see that when putting them together, we were able to see by Western these biotinylated forms, which presumably correspond to interactors of PML only when it is modified by SUMO. And we know that because when we put a mutant form of PML, we don't get this interaction, and we don't get these biotinylated forms. We are also able to follow that in the cell by staining with streptavidin, fluorescent streptavidin. You can see here that when there is the wild type and the SUMO together, they make, we can see biotinylation in these nuclear bodies similar to the PML nuclear bodies, but not when we use the SUMO-related SEBBM form. And by mass spectrometry, we were able to detect many interactors of the SUMO-related PML. The technique was actually proved to be very sensitive and very specific, because we were able to identify specific interactors, for instance, in this case, using P53. When P53 is SUMO-related by SUMO2 or SUMO1, it has a different set of interactors. When P53 is ubiquitinated or is SUMO-related, also has a different set of interactors. So we think that is very interesting to study the interactors of your protein of interest. If anybody is interested in the audience and wants to know more about it, just please contact us, and we will be happy to help if you want to check your protein of interest. And Of course, we had to check SAL because that was our protein of interest in this case, and is the responsible of the Towns-Brocks. And we applied this SUMO ID. SAL is actually, and the whole family is known to be SUMO related. And we actually got a number of factors that they were actually not a lot, but they were very interesting because some of them correspond to the NORD complex that is known to interact with the N-terminal part of SAL, and some of them actually they correspond to Wnt signaling pathway, which also is known to interact with SAL1. So they make sense. And now it's also something that we still didn't explore, we didn't have time to explore further, but we would like to know whether this interaction with these factors, they actually it does depend on the simulation of SAL. So in addition to this dynamic and low abundance thing, another important point in this modification by the ubiquitin likes is that it's very specific. I mean, you might say that all the proteins that get degraded, so it must be a very inspecific pathway, but not. I mean, there are different specificities that actually this is given by the E3 ligases. And to understand, and this is a long important question in the field, is how to detect specific targets for specific E3 ligases. This is a very important question in the field that there have been a number of different strategies that they have been developed, and we believe that we also can contribute to that by the creation of this BioE3 strategy, which is a strategy to identify bona fide targets of specific E3 ligases. But I will not tell you anything about it because Laura Merino later on is going to give you all the hints. That's also part of the project of Ori, the PhD project of Ori and Laura in the lab. So I will stop here, and just as a summary, I told you about developmental disorder, Towns-Brocks syndrome that gives rise to this rare disease, and is based on this truncation of the transcription factor SAL1 that comes out of the nucleus and start making messes when it is in the cytoplasm by interfering with the formation of the cilia, and how LUSP1 centrosomal cytoskeletal protein might be involved in this regulation. And that is a proteostasis situation here by the reduction of LUSP1 in the Towns-Brocks cells. And how to be able to study these two different processes, we develop along the way different strategies like the SUMO ID to identify interactors of the proteins when they are modified, and BioE3 to explore specific targets of E3 ligases. Just the important slide here, the thanking my group, my present group, and also past members that they have contributed to the different aspects of these projects. Special thanks to the parents of children with Towns-Brocks that they donated samples for the study, and also members of bioGUNE, for instance, proteomics facility, and also other collaborators in bioGUNE and elsewhere. And if I have a second, I want just to give you a hint of what we do up here in close to Bilbao. We have this apps club. These are groups that we are interested in the ubiquitin and ubiquitin likes and anything related to ubiquitin. We are different groups, for instance, the group of archives that we have now, Laura and Natalia Martin that will speak here, the group of Malo Martinez Santar and Teresa Cardoso that they already had an elevator pitch talking about Neddylation and liver, the group of Asís Palazón that we have with us Borja Jimenez for this session, the group of Hugo Mayor at the University of the Basque Country that we have a representation of Juanma Ramírez, and the groups of Jose and Nerea Orsinalde. And with that, also thank other networks in which we have been involved, national network called UbiRed, and also this COS ProteoCure for anybody in the audience interested in modifications and proteostasis regulation. You can join ProteoCure, and it will have interesting aspects like meetings and funding for stays, etc. So yes, drop me an email if you're interested, and I can tell you how to do it. Okay, so with that, I think I'm finished, and I can stop sharing my screen, not before I thank again Maria and Sara for the organization of these elevator pitch sessions. Is anybody there?
Thank you, Rosa.
No. Okay.
Very interesting talk. Okay, so now it's time for questions. If you have any questions, please feel free to type them in YouTube so we can read them to Rosa. But meanwhile, while we get some questions to you, I have some, if I may start. So Rosa, you mentioned that these E3 ligases and DAPs that are implied, for instance, in LUSP1 stability, since LUSP1 localizes to both the cytoplasm and to the centrosome, do you know how these ligases or DAPs might get there into the centrosome? Is there a localization signal? They are everywhere and just by chance they happen to the ubiquitin or ubiquitinate these target proteins? Any clue on that?
Well, I mean, what we know is that there are some E3s that they localize specifically at the centrosome in some particular moments of the cell cycle, for instance. There are also some E3s that they localize to the pericentriolar material, for instance, or around the centrosomes. So there is, the regulation seems to be more directed towards the localization of these DAPs and E3s. Whether it is a particular signal, I cannot tell you. I don't have idea on that, but certainly that there is a specific regulation in the localization of E3s and DAPs, yes. There are some proteins that it has been located the localization signal towards the centrosomes or other parts, but I'm not sure that's conserved in the E3s or DAPs that they have been identified at the centrosome.
Okay, thanks. Anybody else has a question? I don't see it. I have another question. So what about, you mentioned the simulation of SAL1. What about the mutated form of SAL1 that these patients have? Do you have any insights on how this protein might be simulated? Sorry.
Yeah, that's an interesting point for two reasons. I mean, we have seen, for instance, in Drosophila that the simulation is important for the function of the protein. And also in the human homologs, there has been some studies saying that the simulation also might be regulating their transcriptional activity, for instance. The thing is that the major site that has been described for SAL1 is in the C-terminal part of the protein. So the simulation sites, in principle, they are not located in the N-terminal part. So we don't have any evidence that the N-terminal part is simulated. But what we see in the N-terminal part are three SUMO interacting motifs, which are these small sequences that are able to interact with SUMO. We have been trying to see whether these sequences are actually functional or not. Still, we are not 100% sure that they actually do bind SUMO. The binding of the SIMs with SUMO is normally very low affinity, and it's hard to detect. So far, we don't have any evidence that they are there. But it's very intriguing that this N-terminal part contains these three SIMs, which might be related either probably with the interaction with other simulated proteins, and may or may not have anything to do with the transbrox. But the fact is that they are there. This N-terminal part has many interesting features. It has like an atypical SIM finger that is very unknown. We don't know what it is about. It has a region rich in polyglutamine that is involved in interaction with other proteins. It has these three SIMs that are still very unexplored. And also has the very N-terminal part is a sequence that is the one that interacting with the NORD complex. So it's full of things, it's full of features. We have to explore that more in detail.
Okay, thank you for the answers. Yeah, I think it's time to move to the next speaker, which is me.
Okay. Okay. Thank you, Laura. Thank you, everybody, for listening. And yeah, go ahead, Laura.
Okay, I will share my presentation. Do you see it correctly?
Yes.
Okay. Okay, so
but Laura, we see it in the presenters mode. You might want to change.
Sure. Second. Here it is. Okay, so now I'll put the presentation mode. Now you see it, right?
Right. Perfect.
Okay, so good afternoon, everybody. As they already introduced me, I just want to say that I work in the Arcaich lab in CIC bioGUNE, where we study the mechanism of prostate cancer metastasis. So first of all, why do we study prostate cancer? It's because it's responsible for many deaths, since it's the cancer with the highest incidence among males in Europe. And you might know that the prostate is a male gland that is located just behind the bladder, and its main function is to produce the seminal fluid that will nourish the sperm. So prostate cancer develops when prostate cells start dividing without any control inside the gland. And there are two potential outcomes or scenarios. One is highly curable, so localized prostate cancer, the cells remain in the prostate, but we are focused and more interested in a more aggressive version of this prostate cancer, which is responsible for the high mortality because prostate cancer cells invade distant organs such as bones or lungs, among others. So basically, when we started this project, we had two goals in mind. One was to select for crucial processes or genes that might lead to metastasis in prostate cancer. And once we have those candidates fixed, we might like to know what's the role in prostate cancer. So get candidates, we basically use two different approaches. One that I call that is previous expertise based, and Rosa already talked about it. But when I was in her lab, in Rosa's Barrio lab, we were studying this cellular antenna that every cell in your body has, which is called the primary cilia. We were studying it in the context of a rare disease called Townes-Brocks syndrome, but it was very clear to us and many others that this antenna might have an important role in in cancer in general. But particularly, we were interested in checking what was the role of this primary cilia or cellular antenna in the prostate cancer. So you may know that in the lab, we have available a prostate cancer mouse model that when we mutate 2 copies of this P10 gene, mice after 6 months develop prostate cancer localized, and they will never lead to metastasis. They will never have metastasis. So we wonder what happens when we knock out or we deplete these prostates in the mice from the primary cilia. Does the prostate cancer become any different? Better prognosis, worse prognosis? We didn't know, so that's what we did. We generated a new mouse colony where we specifically depleted or removed this antenna, this primary cilia, in the prostate of the mice, and we follow them up over time to see how prostate cancer progresses. So what was very interesting to us, from the first moment we saw that these mice without the cilia in their prostate were dying much more frequently and faster. And furthermore, as you can see here in this graph, from 8 months on in red, those mice that had not ciliated prostates were developing a more aggressive version of prostate cancer. As you can see here, the signal in the prostate of these mice, this is tumor mass, is significantly increased compared to mice that have still cilia in their prostates. But even more interesting than that was the fact that these mice, although with cilia, so the double P10 knockout that I mentioned, they don't metastasize. The mice without the cilia, 33% of these mice were developing now metastasis in the lymph nodes at 6 to 8 months. So basically, the conclusion for this part is that we made a new mouse model for prostate cancer that now leads to metastasis to the lymph nodes, and it seems that the cilia is instrumental and very important in mediating this process. So it seems to have a protective role, and when we can deplete this primary cilia, the prostate cancer progresses much faster. But then we also, and that's what I'm talking here, we also develop bioinformatic screening that led us to the selection of different candidate genes that we thought they were very important in metastasis. So basically, we selected in different databases from prostate cancer patients genes that were either upregulated or downregulated exclusively in metastasis compared to primary tumor and normal tissue. And then by applying stringent filters, we came up with a list of 85 genes that were upregulated in prostate cancer metastasis and 114 genes that were downregulated in prostate cancer metastasis. What was very evident to us when we performed a gene ontology analysis was the fact that protein UFMylation was a very enriched category. And you might be like, us at that moment, what is that thing? I mean, it seems very interesting, but what is UFMylation? So Rosa very nicely already introduced post-translational modifications. This is a very less frequent than ubiquitin and very new for the moment. But it basically changes when it's conjugated to the target proteins. It basically changes the localization, function, stability, or other outputs of these proteins. It is conjugated in a similar manner as ubiquitin. It requires the action of 3 different enzymes. So far, there are only one of each described in literature, and this process is fully reversible by the action of the UFMylases. So what was very interesting is the fact that we found in prostate cancer patients, so patients with metastases, that both the ligase, the E3 ligase, and this UFM1 were significantly downregulated. So we thought it was further to analyze. We checked in our mouse model that when mice displayed prostate cancer, the UFMylation, total UFMylation, also the ligase, and the UFM1 itself were significantly reduced. So we thought, okay, what happens when we play with this UFMylation pathway in the cells? Do they get like more or less proliferative? Does the migration change? These are typical characteristics of cancer. So that leads us to the second objective, which is checking, modifying genetically the pathway and see what happens, how the cells behave. So basically, we use 2 different shRNAs to silence UFM1, so the post-translational modification and the ligase, and see what happens with the proliferation and invasion of these cells. What we observed in vitro is by 2 different techniques is that these cells significantly exhibited a reduction in proliferation, as you can see here in the image, but also in the graphs. So the lack of UFM1 and UFL1 leads to a reduction in proliferation. But what was very interesting to us as well, it was the fact that this lack in UFMylation, it led to an increase in the invasive capacity of these cells by spheroid formation in a collagen matrix. Basically, we saw that by silencing this pathway, we were switching from a less proliferative to a more invasive phenotype, but we wonder how this happens. And of course, the first thing to check is whether some UFMylated proteins were maybe less UFMylated and mediating this phenotype towards a more aggressive phenotype. Okay. So far, for the sake of time, I cannot show you many more results, but we applied, thanks to Jim Sutherland and Rosa Barrio, techniques very complementary, where we found that new UFMylated substrates that are at the core of the metabolism in glycolysis and pentose phosphate pathway that we think might be mediating this more invasive phenotype that we observe in our cells when lacking UFMylation. So why is all this important, these 2 words that we are studying in prostate cancer metastasis? Well, on the one hand, we found that cilia might have a protective role in prostate cancer, and this one can envision that it has a very important therapeutic implication. So one can imagine that we can induce primary cilia formation in prostate cancer patients, and by that delaying or inhibiting the growth of the prostate. And for the second part, I hope I convince you that we think that UFMylation is an important player in metabolism regulation in prostate cancer. And that also leads to a therapeutical window where we might think about increasing UFMylation to stop prostate cancer progressions, or even a prognostic value to know which patients might metastasize depending on how the UFMylation pathway is expressed in those patients. So with this, I would like to thank you all for your attention, the Society for their kind invitation, especially my lab, super nice environmental scientific environment, all our collaborators, a special super big thanks to Rosa Barrio that are helping me a lot in this, our collaborators in Spain and also abroad, and all the funding agencies that make this possible. So if you have any questions, please feel free to ask them now. Thank you.
Thank you, Laura. Very nice presentation. Okay, you go ahead, Maria, to moderate. Yeah.
Okay. It's okay if you prefer to moderate. I don't care. Maria is not here, so you can moderate
yeah,
the
of course.
questions.
No,
Perfect.
it's fine. You can go ahead. No problem. Yeah. You can go ahead if you want to moderate. That's fine with me. That's okay. Well, anyhow, either way, I can start. I can start asking. Did you see any relation with the euphemylation and cilia formation?
I think there is an important, another whole project can go on that. I think Jim, that is going to be in the roundtable later, as he is exploring that part. But we think that euphemylation might do something to the cilia. It might even localize there. And there are, some phenotypes that we see when euphemylation pathway is altered that have something to do with the ciliopathies, where the cilia is also functionally or structurally altered.
I have another question. Was UFM the only pathway that you saw enrich or diminish significantly?
No.
Okay.
No. So the gene ontology, we had, as I said, 85 genes and 100. So in total, 200 genes that were altered in prostate cancer metastasis by applying these filters that I mentioned. So another pathway that it caught our attention, but we didn't further explore it, is the actin cytoskeleton was really enriched, integrins pathways. Of course, for the sake of time, the projects have to focus in one of the aspects, and we decided to go for euphemylation, but I think there are very interesting things to check in this actin cytoskeleton regarding prostate cancer metastasis.
But not any other of the ubiquitin likes.
Oh, you mean, no, we didn't find anything like a posttranslational modification signature. No, we didn't find. But we applied very stringent filters. You can, these databases from prostate cancer patients are like very broad, contain a lot of information. You can play around with filters you want to apply. We want it to be very stringent, so only like a very big fold change, very significant, very consistent among the databases. So I didn't explore to lower a little bit the filters and then try and see which other post-translational modifications players might be altered.
Thank you, Laura. So I think Natalia has some question and then Borja.
Hi, Laura, very nice talk. Regarding your mouse model, when you delete or you deplete from cilia, so you observe more metastases. But as long as I know, the cilia will be related more with the division of the cell, right? How do you envision that this cilia depletion will get or give an advantage to the only metastasis? Do you think it's only because the cells proliferate more, or because they acquire more invasion or moving capacities?
Yeah, that's a very important point. My hypothesis is that it plays both roles. On one hand, it increases the division of the cells. When we deplete the cilia, of course, the cells start dividing more. We get bigger tumors in these mice, and therefore the chances that these cells are released from primary tumor and go into the blood system and therefore metastasizing is higher, right? But we also found that these non-ciliated cells have a more mesenchymal phenotype, that they don't localize anymore in the basal part of the prostate, but they kind of migrate towards the lumen, so they can go into the bloodstream. So, it seems that they have also more migratory phenotype, although we are still characterizing it.
Thanks.
So Borja?
Hello, Laura. Very nice talk. I was surprised about your phenotype about in the SH you do less proliferation and more invasion. And I was wondering if have you seen any other case that have a similar phenotype in any other type of research that could help you explain why this is happening?
Yeah, also very good points, all of you. So yeah, we were surprised. We were expecting that when you silence this pathway, because cancer is about proliferation, you get more proliferation in these cells. But this is very consistent. We are sure that this is happening, and it makes kind of sense. Well, just for citing ourselves, when we knock out LUSP1, that Rosa just talked about it, this protein that localized to the actin and the centrosome, we also got that phenotype. We got less proliferation and more invasion. And those mutations in LUSP1 are also present in cancer patients. But on the other hand, you have to imagine that the metastatic process is a very complex like transition of states from in the primary tumor, the cells are they are proliferating. In the blood system, for instance, the cells don't proliferate that much because that's not the ideal environment they want. And then once they reach the metastatic niche, they want to proliferate to induce the metastasis. So they grow again. So there is a switch. There is this known switch from a non-proliferative to a more metastatic phenotype that is known to happen, for instance, in the blood system. And this makes sense in the context of UFM because we found these genes to be altered only in metastasis. So maybe this is a status of the cell when it wants to invade, but it doesn't want to proliferate.
Okay, thank you.
Okay, I think we have time for like one more question or so. There is one in the, or two very quick. There is one in the panels. Is Veronica, do you want to let me just open?
Oh, yes. Can you hear me?
Yes.
So thank you so much, Laura. Very nice talk. So my question was very similar to Natalia's question and was about this maybe side effect of EFT, 88, because maybe removing it, you have side effects. For example, this protein is also related to autophagy, and maybe, you have this phenotype because maybe also autophagy and other biological processes are compromised.
So, also very, very good point. Yes, I mean, IFT88, you want to say something? Sorry.
Oh, no, just briefly. So I don't know if you're thinking maybe I know that it's very difficult, and also if you're working with mouse models, it's very difficult to get maybe another model removing another centrosomal or ciliar protein, just to have the same phenotype, but with different protein, and see if you have the same effect.
Okay, so yes, you're right. IFT88, on top of having a very important role in cilia formation, it has a role in autophagy, in cell division, and all other stuff. What you might expect from knocking out IFT88 regarding cell division is that the cells don't divide that well, and we see the opposite. So that makes me confident that is more than just the cell division role of IFT88. What we are seeing here, I think, is more cilia related. Generating a colony will be super interesting. For instance, we could deplete another gene called KIF3A, more specific to the cilia. Although I have to tell you that this colony formation, so I started this colony in 2019, and now we are starting to see, the phenotypes. Yeah. It takes like 2 years to 3 years. So we are now in contact with a guy in France that is doing a drug screening, searching for drugs that alter primary cilia formation. So that will be, I think, a good way to go, using one of these drugs to validate our phenotype in prostate cancer.
Yeah, and also because maybe you avoid this, chronic situation, which you deplete completely something.
Exactly.
So fantastic. Very nice. Thank you so much, Laura.
Thank you, Vero.
Okay, I think, is there any other question there in the panel? If not, we should probably proceed with the next speaker, Laura. So you take control of the hearing.
Sure. Okay, so our next speaker, is my pleasure to introduce her, is Laura Merino. So she obtained her bachelor degree in biochemistry and molecular biology, and also her master's degree at the University of Basque Country, where she graduated with honors. And now she's currently doing her PhD at Rosa Barrio lab, working on posttranslational modifications and the development of biotin-based strategies. So, Laura, whenever you're ready, you can go.
Okay, can you hear me?
Yes, you can, but okay, put the presentation. Yeah, no, yeah.
Okay, now, no?
Yes.
Okay, so thank you, Laura, for your kind introduction. So in these 5 minutes, I'm just going to give you a glimpse about one of the projects that we currently have in the lab, which consists on the development of biotin-based methodologies to study ubiquitin targets. So as Rosa has said, post-translational modifications by ubiquitin happen in a 3-step enzymatic cascade. But in this talk, I just want you to focus on the E3 ligases, which are the ones that we confer substrate specificity to the process. So there are more than 600 different E3 ligases, and each of them has their own particular subset of target proteins. So to decipher this E3 substrate pairing is one of the major challenges in the field. So for that, I want to present to you now BioE3, which is a biotin-based technology that will allow us to identify targets of E3 ligases. It relies on 2 main elements. First of all, we have our E3 fused to the B-ray enzyme, and on the other hand, ubiquitin or ubiquitin-like proteins fused to the biotin acceptor peptide habitat. So, in the presence of a target protein, the E3 will recognize it and promote its modification. And at this point, the B-ray and the habitat will be in close proximity. So biotinylation of the peptide will happen, and in this way, we will tag our substrate that is modified. So we can later on purify it using streptavidin beads and identify them. So to test our methodology, we chose as proof of principle RNF4, which is a STABL, which stands for Sumo Targeted Ubiquitin Ligase. It interacts with simulated target proteins through its sumo interactive motifs and promotes its ubiquitination. So for testing this BioE3, we generated 2 different mutant versions of TC3, one lacking the same domains, so it cannot interact with the simulated target proteins, and a CE version, which is catalytically dead. So first of all, we test BioE3 by Western blot. For that, we transiently transfected the B-ray RNF4 construct into cells that express ubiquitin fused to the habitat. And we observed that we were able to accumulate biotin-related proteins in the wild type in comparison with the mutant versions. This suggested that BioE3 specifically labels ubiquitin targets of RNF4. Then, by confocal microscopy, we could observe that the B-ray RNF4 construct had a nuclear localization, but only in the wild type we could see biotin-related proteins, also nuclear. Last questions that we asked ourselves was, can we identify an already known target of RNF4? And we chose PML. PML is ubiquitinated by RNF4 upon ATO treatment, which makes them to be degraded by the proteasome. We perform a BioE3 experiment. We treat the cells with ATO and the proteasome inhibitor MG132, and we purify the biotin-related proteins by streptavidin pull down. And as you can see, we were able to identify PML as a substrate of RNF4, but only when treating the cells with ATO and MG. And also by confocal microscopy, we could observe how these 2 treatments promoted the formation of PML nuclear bodies, recruited the B-ray RNF4 construct, and promoted biotinylation of target proteins at boost losses. So finally, we performed a big-scale mass spectrometry experiment when we were able to identify 259 proteins enriched in the wild type in comparison with the CA, which we consider that are putative targets of RNF4. Among them, we found components of PML nuclear bodies and proteins related to the ubiquitin machinery. And we also compared the wild type with the delta scene. And in this case, we identified 280 proteins. And when we compared the 2 datasets, more or less the 80% of the proteins were served by both of them. And those we think that are sumo-dependent targets of RNF4, suggesting that the sumo-sim interaction is the main mechanism by which RNF4 recognizes their target proteins. So just to conclude, BioE3 specifically labels and identifies target proteins. By confocal microscopy, we can see the subcellular localization of those biotin-related proteins. We can also identify stimuli-dependent targets, and also BioE3 gives us some clues about the main mechanism by which the E3 ligases recognize their substrates. So overall, we think that BioE3 is a powerful method to identify substrates of E3 ligases. I hope that I've convinced you in these 5 minutes, and well, just thank the lab, the collaborators, too, for listening to it, and feel free to ask any questions now.
Okay, Laura, very nice talk. Thank you. So again, feel free to ask any questions if you want. We have already a raised hand by Natalia. Go ahead, please.
Hi, Laura, very nice. I was wondering, since PML bodies and the simulation are very dynamic processes, have you tried, I don't know if it's possible even, to monitor in live this by microscopy, see how the processes are being occurring like in live, like with a movie or something like that?
Yeah, I mean, we had that in mind, but as I said, it's pretty difficult to perform. But yeah, it was something that we were interested in, but we haven't performed it yet.
Okay.
Okay. I also want to ask you something. So can you comment on the specificity? How specific is this technique? I mean, do you think that you have to, like, of course you have to validate, but how important the validation is? The candidates that you get, you think they are very like strong candidates, target proteins? Can you comment on this?
Yeah, well, for the sake of time, I simplify a lot, like the basis of the technique. We introduce a lot of variations to this B-ray habitat system to make it even more specific. And I mean, we really think that it's a quite specific technique. Obviously, maybe there is, we identify some proteins that are not targets, but I think that it's very specific. And if then you have like a very interesting hit, obviously validation is needed, but really believe that we are reaching good levels of specificity.
Okay, thanks. We have a question from Juanma Ramirez. Juanma, go ahead, please.
Hello, Laura. Thank you very much for the talk. I have just a fast question because you have presented this strategy, and I was wondering, have you tried, or are you thinking to try to combine the bio E3 with different ubiquitin mutants to see whether you can enrich substrates that are specifically modified with certain ubiquitin chains?
I mean, we haven't think about that, but yeah, it may be. Yeah,
I think
sorry,
that.
I mean, I can't hear you.
Sorry. Yeah, I mean, I think that it might be interesting to try this one just to try to decipher which type of ubiquitin chains are formed by some specific E3 ligases.
Yeah, I mean, then with the proteomic analysis and these things, we add this diglycine modification. So as we obtain ubiquitin there in the list of target proteins, we can infer a little bit the chains that are formed. And also then we make geo-analysis, and sometimes we show enrichment of specific ubiquitin chains. But yeah, it may be interesting also to try it.
Thank you.
One last question, Laura, if I may. I saw you have many candidates there for RNA4. Are you planning on, although I don't want you to scoop it, but are you planning on working on any other E3 ligase other than RNA4?
Yeah, I mean, we try this methodology with some more E3 ligases. We try to make to use like organ-specific E3 ligases like mitochondrial. We also try with some E3 ligases that are not very well characterized, just to see what what can we obtain. And well, all these and also RNA4 are ring-type E3 ligases, which they catalyze a direct transfer of ubiquitin from the E2 to the substrate. But there are more classes, and we implemented this technique to HEC types, which there is like a intermediate transthiolation step, and introducing some modifications. So yeah, I mean, I think that we tried in overall like with 5 different T3 ligases, and it worked really good. So we're very happy.
Okay. So if there are no any other further questions, we can, I think, move to the next speaker of today. So I'm very happy to introduce to the audience Borja Jimenez Lasheras. He graduated in pharmacy at the University of Basque Country back in 2017, the only pharmacist among the panelists. And after a broad internship here in Boston, he worked as a lab technician in the immunometabolism and cellular signaling lab at CNIO in Madrid. And then he started his PhD in the group of Asís Palazzón at CIC bioGUNE, where he is now studying the role that Neddylation might have as a regulator of immunity. So whenever you're ready, the floor is yours, Borja.
Well, thank you, Laura. Hello, everybody. Today I'm going to introduce my PhD project, which is focused on Neddylation as a relevant regulator of CD8 T-cell mediated anti-tumor immunity. So, as you all know, Neddylation is a ubiquitous post-translational modification, which is based on a ubiquitin-like protein called Ned8. Ned8 is covalently bound to substrate proteins, and in some cases this means degradation of these proteins, and in other cases means the stabilization of these proteins. Since my group works on anti-tumor immunity, we saw that Neddylation is upregulated in some tumors. Actually, there's since 2007, there's this Neddylation inhibitor called MLN4924, which is an anti-tumoral candidate and is being evaluated in clinical trials. But we were wondering what's the effect of inhibiting Neddylation in our immunity, especially in CD8 T cells. So first of all, I will briefly introduce the Neddylation cycle. It's very similar to the ubiquitin cycle. There's one E1 activating enzyme called NAE, which activates Ned8 and transfers Ned8 to the E2 conjugating enzymes, which will transfer Ned8 to the E3 ligase. And this complex of E2 and E3 with other proteins will finally conjugate Ned8 to the final substrate. There are two main ways in which Ned8 is bound to the substrates. One is to coulins, which are currently 80% of the known substrates of Ned8 are coulins, and Ned8 is necessary for their function. So when a coulin gets Neddylated, there's a conformational change, and thanks to this change, coulins are able to ubiquitinate proteins, and the cell has a correct protein degradation. But there are other substrates which are the most interesting right now, which are also Neddylated through other E3 ligases. And in this case, the E3 ligase can take Ned8 and put Ned8 into the final substrate. So our hypothesis is if Neddylation is necessary for CD8 T-cell mediated anti-tumor immunity. And therefore, first of all, we have some models for Neddylation inhibition. What we did first is we checked that Ned8 is expressed in both human and mouse CD8 T cells, and that we activated these T cells in vitro. We saw that Neddylated coulins were upregulated. Therefore, the Neddylation pathway is upregulated. And we also checked that in vivo, where CD3 T cells express Ned8 inside the tumor, in the tumor microenvironment. Given this, we have a genetically modified mouse which lacks NAE, the first enzyme of the process, exclusively in T cells. And we made some in vivo experiments with these mice. First of all, we saw that when we injected a cell line subcutaneously in the mouse, we analyzed the tumor infiltrate and we saw much less T cells in the mice that lacked NAE, exclusively in T cells. And we also saw that CD8 T cells that lack NAE have a less tumor control, and the survival rate of this mouse is much worse. So given this, we think that NAE is essential is relevant at least for the CD8 functionality against tumors, but we want to try if we can improve the CD8 functionality by modulating other steps of Neddylation. We know that the first step is quite important, but there are other E3 ligases that they have been documented to have Neddylating capacity. There are other substrates that it is known that they get Neddylated, and we are trying to play with this in order to modulate Neddylation and have a beneficial effect in the CD8 T cell. So given this, I would like to thank my lab mates from the Cancer Immunology and Immunotherapy Lab, to my PI Assis Palazón, to deliver this lab, and to Aswin, who generously donated the NAE knockout mice.
Okay, thank you, Borja, for a very interesting presentation. Does anybody have any questions? Yeah, I think we have a question from Ori Barroso. Ori Barroso. I don't know if he can open the microphone or he just have to.
Yes, I open it.
No?
Okay. Yes, Ori, we hear you.
Okay. Borja, nice presentation. Thank you. So you said the Neddylation of the coulins is enhanced when those T cells are activated, right?
Yes.
Is the ubiquitination activity also enhanced? So do you have some data suggesting that somehow they are more Neddylated but also more active?
Actually, we are not completely sure about that, but what we have seen through Western blot is that that CD8 that lack Neddylation somehow have the ubiquitination enhanced, and maybe it's been described that when there's no NAE, there's an alternative pathway that uses ubiquitinating enzymes. So somehow we actually think that when Neddylation is decreased, ubiquitination might be increased.
Okay, thank you.
Okay, I think Tim is raising his hand. Tim, go ahead, please.
Hello, Borja. Can you hear me?
Yes.
Yes. Thanks for your talk. So you say when you knock out the NAE enzyme, you have less T cells infiltrating the tumor, right?
Yes.
Are there any assays that you can do in vitro with T cells that by using the NAE inhibitor, the drug, you can get some kind of clues on why those T cells are not infiltrating? Any kind of assays like cell biological assays or anything like that?
Yes. Actually, we have this doubt about. We find less T cells in the tumor microenvironment, but actually we are not sure of if they arrive and they die or they do not proliferate, because we see less proliferation in NAE knockout CD8 T cells, or either if they are not able to arrive. And actually we are thinking about doing migration assays, because the problem might be that they are not able to arrive to the tumor.
Okay. Okay, that makes
yes.
sense. Okay, thank you.
Thank you.
Okay. While we wait for any other questions, I have one question. Do you have any idea how this inhibitor of Neddylation works? What is targeting in cancer? You mentioned there is an inhibitor now.
Yes. This inhibitor blocks NAE activity, so it should be the effect. It should be quite similar to the mouse. The mouse we have. Basically, it blocks the cycle in the E1 enzyme. However, there's more and more research saying that this inhibitor, it blocks very efficiently Neddylation, but it also has some off-target effects, which actually we see some things that do not replicate exactly from our mouse to the inhibitor. So it seems that it also has a strong anti-inflammatory effect. So it's very efficient blocking Neddylation, but still there are clues to know about other off-target effects, you know.
Okay. And last question, if I may, do you have any clue on the mechanism by which this NAE knockout leads to less infiltration, like the real candidate? Do you think it's a substrate of NAE that is now not being Neddylated, and therefore it affects the functionality of these T cells? So this is in line with Jim's question, but more like deeper into the mechanism.
That's what we are trying to solve. We are trying to get what are the Neddylated proteins in a T cell, because we are not pretty sure. We think that there's a cell cycle, some kind of cell cycle arrest, which has been documented with the inhibitor, and since we see a strong proliferation impairment, we think that there's something occurring in the DNA that is blocking the proliferation, but it's quite difficult to say.
Okay, thank you. Any other questions? Not as far as I see. So thank you, Borja, once again.
Thank you.
So our last speaker of the elevator pitches is Juanma Ramirez. Ramirez, you can start with your presentation while I introduce you. So Juanma studied biology at the University of Basque Country, and also his master was studied there in molecular biology and biomedicine. Then he went for an internship, summer internship to Cambridge, and also another internship of 6 months in University of Tennessee in Memphis, USA. After that, he started working at the University of Basque Country, where he has been enrolled as a postdoc in the ubiquitin world until last year. Congratulations. Well, this year, sorry. Congratulations on that. You become, I don't know how to say it exactly, but associate professor at the UPB. Is that correct?
Yeah, I don't know if that's the word, associate professor, but yes. Thank you very much.
Yes, so congratulations, big achievement.
Thank you.
Okay, go ahead.
Okay, so can you hear me properly and you see the presentation? Okay, so
yes.
Thank you very much, Laura, for the nice presentation. So today I would like to give you a glimpse of what we are doing of the projects we are carrying in the lab at the University of the Basque Country. Our projects are focused on understanding the Angelman syndrome, and for that we're using a ubiquitin proteomic approaches. So Angelman syndrome is a rare neurodevelopmental disorder which is characterized by a severe developmental delay, speech impairment, movement or balance disorders, and in many cases, patients also suffer from epilepsy. The main cause is the loss of function of a ubiquitin E3 ligase in neurons known as UB3A. As they have already introduced, Rosa and Laura and all of them, ubiquitin E3 ligases are in charge of modifying substrates with ubiquitin. Therefore, identifying the target subsets of U3A in neuron could really help on understanding which are the molecular basis of this syndrome. So since 1997, when mutations on the gene were related to the syndrome, many attempts have been performed in order to identify the substrate that this E3 ligase is regulating. Some of those work were based on interaction with the E3 ligase or based on changes in the total protein level of the targets, assuming that once they are ubiquitinated, they are target for degradation. Some other work has been performed with in vitro experiment. However, nowadays, still the identification of potential UE3 substrates in the neural context still remains a challenge. And this is where our labs comes to play, because as Rosa and Laura has already explained, we are also using the BRA technology. We're performing in vivo biotination of ubiquitin because BRA has the ability to put a biotin on a small peptide known as ABITAC. Like this, we are able to label all the cellular proteins that are modified with ubiquitin, and therefore this allows us to very easily fix them up using avidin bits. Once we have isolated, we can identify them by mass spectrometry and identify them. If we combine this strategy with the overexpression of one E3 ligase, we can identify which are the targets of this E3, because we expect that the target of these subsets are going to be the ubiquitinated fraction of these targets are going to be enriched on those samples where the E3 is being overexpressed. And in fact, as a proof of principle, we test that in Drosophila melanogaster, where we overexpress this BRA and the biotinated ubiquitin together with UE3A, and we managed to identify 79 proteins that were found more ubiquitinated by UE3A in Drosophila neurons in vivo. And here I'm showing you a Western blot where you can see how when we overexpress UE3A, there is a huge increase on the ubiquitination fraction of some of its substrate, in this case, Ringo. So having done that, we decided to move it to a more complex animal model, and we performed similar approach in mice. Here we also overexpressed the biotinated ubiquitin together with UE3A in neurons, and in this case we managed to identify 132 proteins that were more ubiquitinated by UE3A in mouse neurons. So among those results, just in those 5 minutes, I would like to share with you a couple of results. One of them is that among the target proteins that we identified as UE3A substrate, as putative UE3A substrate, as an average, we found that most of them has a significantly bigger size as compared to the remaining proteins identified. We still don't know whether this is something with biological highly significance, or is just something related with the technique, or with the fact that many proteins we identified were coming from the plasma membrane. But we found that this result interesting. And the second result that I would like to share you with which we think is really important, and I would like you to be your take-home message from this talk, is that we didn't find any correlation at all between the protein abundance and protein ubiquitination level. What does this mean? That we should not assume that a bigger ubiquitination of substances will lead to their degradation, as it is usually assumed. So this highlights the importance of performing ubiquitin enrichment protocols, such as this one with the BRA, or the ones that Laura has presented before. And that being said, I will ask to finish just thanking you all, and showing you a picture of the lab and all the fundings we have. Thank you very much.
Okay, Juanma, thanks for the very nice presentation. I think Rosa Barrio wants to ask a question.
Thank you, Juanma. How do you distinguish then with this overexpression something that can be and direct, because there you don't know, do you have any special precaution like short labeling times, or are you doing something in particular there?
No, so since this is in vivo, we are not doing all the time the E3 and the ubiquitin are being expressed, and so we don't make short times exposure to the expression, but we usually consider, or we try to get some stringent stringent criteria to consider one protein subset or not. And usually we set up as a threshold that at least the protein has to be twofold times increase in order to consider a significantly more ubiquitinated and therefore as a protected subset. In terms of whether this is indirect or not, from our data, I think it's difficult to distinguish, but then we usually perform independent experiments with the proteins of interest just based on GFP pull-downs or maybe, yes, or GFP pull-downs, and trying to see whether specifically this protein is ubiquitinated by the 3.
And you do all these experiments in the absence of proteasome inhibitors, or?
Yes, so this is in the absence of proteasome inhibitors, yes.
Okay, thank you.
Okay, Jim Sutherland has another question.
Hello, Juanma.
Hello.
I have a question about, so are there any known substrates of UBE3A that through in vitro experiments, they know what kind of chains it's putting on to substrates?
So yeah, there are some experiments and they have tested, many of them suggest that U3A preferentially ubiquitinate proteins with lysine 48 chains.
Okay, because what you were saying is that there's not a good correlation between the amount of ubiquitination and degradation.
Yeah.
So does that, do you think that those are K48 chains that are not leading to degradation?
There might be. So there are some papers that suggest that lysine 48 chains do not necessarily send proteins for degradation, that rather they might perform a conformational change in the protein, so the protein is somehow get inactivated, but it is not degraded. So yes, so I mean there might be other functions of apart from protein degradation.
Okay,
But
okay.
we don't know whether in this so yeah with our data we can guess what kind of proportion of lysine 48 chains we have, but for to know whether the specific subset of U3A are being targeted with lysine 48 chains, we need to perform independent experiments.
Okay, all right, thank you.
Okay, so thank you, Juanma. I think we have to move for the sake of time to the roundtable now. I don't see any further questions. Thank you all. It was a wonderful session, all very interesting talks. And for the roundtable, I would like 2 people to join to this panel. In first place, Natalia. Hello, Natalia.
Hello. Hello, everybody.
Yeah, so Natalia, let me introduce you, studied biochemistry at the University of Salamanca, and then she moved to a very nice city, Dublin, where she performed her PhD, studying how different pathways affect the renal epithelial barrier function. And then in 2010, we had the pleasure to have her back in Bilbao, where she joined the Arcaiz Carazados lab as a postdoctoral researcher first. And then since 2019, also congratulations, she became a research assistant, a very rare but super nice position in the world of science. And she's now leading a line of research on metastatic prostate cancer. So thanks, Nati, for joining us in this roundtable. I will switch now to Spanish, sorry.
Vale.
I forgot. La mesa redonda la vamos a hacer en castellano. Y me gustaría también presentaros a Jim Sutherland. Hola, Jim.
Hola.
Originalmente de Ohio, pero bueno, habla muy bien castellano, así que en primer lugar estudió en la Universidad de Oberlin biología, luego hizo una tesis en la prestigiosa Universidad de Harvard en Boston. Después hizo 2 postdocs muy exitosos, uno en el UNBL en Roma y después en el Cancer Research in UK en London. Y después, desde el 2005, es Associated PI, PI and Associated PI en el CIC bioGUNE en Bilbao, lo que es un placer para todos nosotros que convivimos con él. Bienvenido, Jim.
Gracias.
A la mesa redonda. Entonces, para romper un poco el hielo, vamos a empezar. Podemos hablar de cualquier cosa, podéis sacar cualquier tema que queráis, pero para romper el hielo, yo he pensado que siempre se habla de por qué estás aquí, qué haces en la ciencia, pero me parece también interesante pensar qué hubierais sido si no sois científicos en otra vida, o si no hubierais elegido la carrera investigadora, ¿qué creéis que os gustaría hacer? Si os parece, empezamos con Jim o Nati, que se han incorporado y no los hemos escuchado todavía.
¿Conmigo?
Jim o Nati, sí.
Vale. No, bueno, es que yo tengo una hermana que estaba estudiando medicina y probablemente si no estudio biología, probablemente soy médico, pero también he crecido en una granja de pollos y entonces es posible que también puede ser un campesino, así. Sin más.
Vale, Nati, si quieres añadir.
Pues qué difícil. La verdad que me gusta mucho la docencia. Ahora estoy dando clase en la universidad y me gusta mucho. Pero sí que es cierto que está asociado, doy clase en la universidad en el laboratorio, enseñando las técnicas que hacemos en el laboratorio. Entonces, sigue un poco enlazado. Me gusta la docencia, como dice Jim, bueno, pues también mi vida me ha llevado a hacer diferentes cursos y así. Creo que si no hubiera sido científica, probablemente profesora, ya sea de yoga, pilates o de lo que me hubiera surgido, pero me gusta la docencia en general.
Muy bien. Podéis intervenir el resto cuando queráis.
Okay,
Yo creo que tenemos, perdona, Juanma, que quería añadir algo, ahora sí. Sí que creo que tenemos algo en común los científicos, que aunque no hubiéramos sido científicos, quizá esta motivación, ¿no?, por aprender cosas nuevas y trasladárselo a otras personas, creo que en cualquier profesión, siendo campesino también, Jim o lo que fuera, creo que nos hubiera salido un poco esta vena de buscar cosas nuevas cada día, algo nuevo que hacer y trasladarlo. Juanma, perdona que te corta.
Nada, no te preocupes. Pues, no sé, a mí siempre me ha gustado el tema de diseñar por ordenador y así. Este tema de diseñar cosas con programas informáticos o ilustraciones siempre me ha gustado. Entonces yo creo que igual si no hubiese hecho la carrera de biología, igual hubiese ido por ese lado, en hacer algo de diseño por ordenador o ilustraciones o no sé. Me entretiene, por ejemplo, hacer las presentaciones de PowerPoint, no tanto luego presentarlas, a veces es más esto, pero lo que es hacer el proceso me parece totalmente entretenido.
No sé, en mi caso, sí, en mi caso un poco cojo los comentarios de Nati y de Juanma. Por un lado, eso, que algo que tiene de bueno la investigación o la biología, etcétera, es que cada día es diferente un poco, ¿no? O sea, no existe el aburrimiento en ese sentido porque tenemos diferentes challenges y cada día tienes que enfrentarte a cosas diferentes, etcétera. Y yo creo que eso está bien, porque creo que en un trabajo que fuera más monótono, con menos challenges, me aburriría, acabaría alienada totalmente. Entonces sí, un poco trasladar, si es cualquier otro trabajo, digamos, que suponga así un continuo challenge, yo creo que sería interesante. Y luego, de hecho, trabajé antes de empezar la carrera de biología, trabajé en varias cosas. Y uno de los trabajos que hice, pues es lo típico que acabas el instituto, antes de empezar la carrera vas a sacar un dinero y tal. Y uno de los trabajos que hice fue diseñadora gráfica en una empresa. Durante un tiempo relativamente corto. También estuve trabajando en el bacalao, ahí en San Sebastián, etcétera. O sea, son trabajos para sacar dinero, viendo lo dura que es la vida laboral, especialmente en trabajos como esto del bacalao, que era muy duro físicamente, etcétera, es lo que me animó definitivamente a seguir estudiando. Pero, por ejemplo, diseñadora gráfica era algo que me gustaba. No tenía ni idea, yo era muy jovencita, pero bueno, hacía lo que podía. Y si hubiera tenido a lo mejor otras inclinaciones, si hubiera seguido a lo mejor algún tipo de trabajo por ahí. Me gusta ahora, de hecho, también lo de las figuras. Me gustaría tener tiempo para estudiar más y meterme ahí a ver cómo funcionan los distintos programas, que al final los usas a nivel muy superficial, pero sí, nunca hay tiempo, la verdad, para meterse en detalle.
Muy bien. Laura o Borja.
Hola, pues no sé, yo creo que seguramente seguiría en el ámbito científico, aunque no fuera en ciencia. Yo creo que posiblemente sería ingeniera o algo similar. Pero vaya, que en realidad yo creo que si no fuera científica en biología, sería científica en química o en física, o siempre tirando más por la ciencia. Pero bueno, si ya me lo ponéis tan difícil, diré que ingeniería.
Pues en mi caso, bueno, yo estudié farmacia, entonces yo antes de empezar la tesis trabajé en una farmacia y sí que hablando de lo de la monotonía que decía de Rosa y así, sí que era un trabajo mucho más monótono que el que hacemos ahora todos los días, ¿no? Entonces en ese sentido sí que la ciencia te ofrece eso. ¿Yo qué habría hecho? Pues ahora no sé decir, seguramente a lo mejor profesor de instituto o así también. Eso yo creo que me habría gustado bastante.
Muy bien. Veo que el factor común es ciencia-arte, que arte también está íntimamente ligado a la ciencia, la creatividad. Así que la manzana no cae muy lejos del árbol, veo. En mi caso, yo vengo de una familia de agricultores y me he pasado muchos veranos y septiembres cogiendo patatas y uvas. Las patatas eran realmente duras, aunque tengo que admitir que alguna vez en la ciencia me he planteado si volver a coger patatas, dependiendo de la fase. Pero sí, el factor común es que los científicos llevamos todo al método científico. Incluso cuando estoy cogiendo uva o patatas, intento buscar la n igual a 3 experimentos, esta fila funciona mejor que la otra. Bueno, y creo que es una buena señal, ¿no? Que significa que trasladamos nuestra pasión también a la vida personal. Entonces yo diría que agricultora o profesora, cualquiera de esas dos me valdría, pero estoy muy feliz de momento por aquí. Vale, vamos a meternos un poquito más en harina, en cosas serias. Se me ha olvidado comentar que cualquiera de los participantes puede hacer preguntas e intervenir en la mesa redonda si lo consideran de interés. Pero creo que un problema muy importante, y muchos de aquí probablemente estéis de acuerdo, es que en la ciencia, en el sistema científico y sobre todo en el español, hay una falta notable de posiciones o de puestos de trabajo para esos postdocs, o sea, esa gente que ha hecho la tesis, pero que no quiere ser PI. No hay muchas oportunidades. ¿Cuál es vuestra visión de esto? ¿Cómo lo mejoramos? ¿Qué hay que hacer? Y le doy la palabra a Nati o Jim otra vez para hacer el círculo.
Vale, yo si quieres empiezo porque como has dicho antes, pues yo he tenido la suerte de ser esa figura justo. Yo sí tenía claro desde el principio que me gustaba mucho la ciencia, pero no me veía en la posición de PI. No quería dejar el laboratorio, me motiva mucho más estar en el laboratorio haciendo experimentos que escribiendo proyectos, pidiendo grants. Bueno, que no todos tenemos las mismas inquietudes, eso es verdad. Y me costó mucho. Me acuerdo que fue, de hecho, Manolo Serrano el primero que me comentó que en el CENIO había este tipo de posición. Y bueno, pues tuve la suerte de que Arcaiche, cuando consiguió la RC, pudo negociar mi estabilidad y por eso estoy como Research Assistant, pero sí que creo que es algo muy difícil de conseguir en España, lo cual me parece tremendo porque hay un cuello de botella que hoy en día yo le veo difícil solución. O eres PI o digamos vas un paso más atrás y quizá puedas trabajar de técnico de laboratorio, pero no se te va a reconocer toda tu carrera, o te tienes que ir a docencia, como estamos viendo con muchos compañeros, docencia, industria, empresa privada. Entonces es una pena porque toda la formación que llevamos acumulando, tanto para nosotros es una pérdida como para el grupo en el que estamos, que también podemos aportar mucho siendo postdocs veteranos, digamos. Entonces, no sé la solución, no sé, o sea, obviamente viene de la inversión que se hace y de la apuesta y de dejar un poco al lado la idea de que porque seamos científicos nos tenemos que estar constantemente moviendo, ir a otro país, ir a otro laboratorio. No tiene por qué ser siempre beneficioso.
Muy bien. ¿Algo que añadir? ¿Alguien?
Yo creo que también es un problema político, porque
Totalmente.
yo creo que no es un problema único en España, es todo el país es lo mismo, que hemos formado muchos estudiantes, hay mucho conocimiento, pero hay pocas plazas. Y entonces tienen que repensar, cambiar el modelo, que no es tan como pirámide, que tienes un PI y los otros puestos. Tenemos que crear puestos nuevas que no son los PIs, son gente mayor que investigación es su trabajo. Y entonces hay un frase en inglés que dicen research de blue skies research. Y la idea es que investigación es algo que es un lujo, que cuando hay poco dinero es la primera que corte, es este tipo de investigación, porque el público o los políticos piensan que no es necesario. Y esto no es correcto. Los mismos políticos saben que conocimiento es como oro, y todos los demás, todos los productos que salen de investigación, de farmacéuticos, de tecnología, biotecnología, mucha investigación en agricultura, viene de investigación de blue skies. Y si deciden cortar esto, es contrario. Y entonces, bueno, porque es una cosa muy económico, yo creo que tienen que pensar y cambiar y poner más puestos de este tipo. Porque hay mucha gente con mucho conocimiento, mucho talento que estamos perdiendo. Y es una pena, porque, bueno, hemos formado bien y luego se sale.
Sí, yo solamente apuntar, estoy de acuerdo con lo que se comenta, que, bueno, en principio que la gente vaya hacia industria, etcétera, no está nada mal. De hecho, se necesita para que haya también otras posibilidades, otras posibilidades, y que haya una industria en la que se haga buena investigación. O sea, eso no está mal, lo que pasa es que me parece que la oferta todavía sigue siendo muy, muy baja. Pero bueno, igual nos podrías, por ejemplo, Laura, Juanma, vosotros, ¿cómo lo veis? Porque estáis precisamente en ese momento ahora. Por ejemplo, Nati tiene una situación más estabilizada, ¿no? Con este puesto que es tan difícil de, como ya he dicho, de conseguir. Borja y Laura todavía están muy al principio de sus carreras. Y vosotros, Laura Bozal y Juanma, estáis en el momento crítico, ¿no? De cómo asentarse, etcétera, ¿no? quizá vosotras tenéis la visión más directa de precisamente este problema.
Sí, yo hasta hace poco estaba contratado por un artículo que es el de la ley de la ciencia, el artículo 22, en el que, bueno, tienes cierta como cierta categoría o así en lo que es el ámbito científico, pero solo te permite estar contratado bajo ese tipo de contrato durante 5 años. Una vez que se acaba ese periodo, eso, o accedes a una Ramón y Cajal o un Ikerbas, o es muy difícil encontrar un trabajo estable. Y hasta ahora pues estaba mediante contratos asociados a proyectos que además de que son muy temporales muchas veces, porque esa es otra cosa, que a veces el laboratorio tiene dinero para comprar muchos aparatos así, pero para personal a veces no está tan disponible, pues claro, estás en un momento de inestabilidad con ese tipo de contratos. Y no solo tú, sino el laboratorio también. Ahora con la nueva ley no sé cómo está eso, igual ha cambiado y eso seguro que hay alguien que sabe mejor que yo, pero hasta ahora, pues, al estar contratado con un contrato tipo asociado a proyecto, a muchas convocatorias el laboratorio ni siquiera podía utilizar tu currículum para pedir otras becas, sino que como estás asociado a un proyecto específico, no pueden meter tu perfil, por lo que eso perjudica también al laboratorio como entidad. Entonces yo tuve la suerte de que justo en el departamento ha habido bastantes jubilaciones y he optado a una plaza y he sacado una de las plazas de profesorado, pero pues si no, la verdad es que no sé qué se ha pasado, porque claro, ya llega un momento en el que tampoco puedes acceder a ciertas becas por edad y entonces, pues no lo sé. No sé cuál es la solución, yo creo que tiene que venir desde arriba. No sé si es del gobierno o desde las instituciones que asignen ciertos dineros para ciertas categorías. La verdad que no sé cuál puede ser la solución.
Por alusiones, estoy de acuerdo con todos vosotros. Creo que tiene que venir desde arriba, pero también creo que tiene que venir desde abajo. Es decir, somos muy buenos en quejarnos, pero somos muy malos en actuar. Y creo que se tiene que oír que hay un problema general, la sociedad lo tiene que escuchar, igual no les interesa tanto, pero al final hacer ruido siempre ayuda. Creo que las organizaciones privadas son más conscientes, por ejemplo, la Asociación Española contra el Cáncer, que yo soy becada por ellos, en mi etapa postdoctoral es súper consciente de ello y ha creado como unas plazas, bueno, unas becas, perdón, para esa transición o ese periodo entre la tesis y el PI, si al final lo decides así. Pero son muy puntuales y muy escasas. Sé que ellos están haciendo también desde la asociación presión en el gobierno, pero es una cuestión de dinero, como estáis diciendo. Estabilizar a una persona en esa escala, vamos a decir, es un coste muy alto. Los laboratorios pequeños, desde luego que muchos de ellos no se lo pueden permitir. Solo los laboratorios grandes, pero el sistema público podría permitirlo, generar como unas oposiciones, pero creo que no se ve. Y no se ve por 2 razones, porque no lo quieren ver, la primera, pero la segunda es porque no nos movemos lo suficiente. Y soy consciente de que es mucho esfuerzo y no sé si se conseguiría mucho cambio político, pero desde luego que tenemos que hacer algo también los investigadores. No sé, Laura y Borja, cómo lo ven. Hoy igual no lo tienen claro, pero igual podéis aportar vuestra opinión.
Igual Laura y Borja nos pueden decir si ellos se ven en un futuro eso, teniendo su propio grupo o qué es lo que tienen en la cabeza o qué ideas.
Al hilo un poco de lo que decía Laura, yo sí creo que la sociedad sí que sabe, ¿no? Pues que la ciencia, que es mucho trabajo, que es muy sacrificado, que en muchos casos no está bien pagado. Yo creo que todos esos mensajes sí que están, pero es verdad que este mensaje concreto de los puestos después del postdoc, algo permanente, yo creo que eso no se conoce. No se sabe esa reivindicación. Entonces ahí sí que estoy con Laura bastante, la verdad. Y bueno, en mi caso, yo creo que encajaría en el perfil de que tampoco quiero ser PI. Eso de momento lo tengo bastante claro.
Nadie lo quiere, nadie lo
quiere.
Qué
qué mala imagen damos los PIs, la verdad.
Nadie lo quiere. Al principio todo el mundo dice lo mismo y mira, aquí están.
Pero bueno,
Bueno, pues a mí sí que me gustaría ser PI ahora mismo. O sea,
Muy
en
bien,
un
Laura,
futuro.
muy bien.
Pero bueno, o sea, también soy consciente de que luego la vida da muchas vueltas y que dependiendo de la situación en un futuro, mi opinión pueda cambiar. Pero sí, definitivamente creo que es necesario alguna posición intermedia que dé cierta estabilidad en el ámbito científico, porque al final somos personas y también necesitamos esa estabilidad para tener nuestra vida.
Vale, pues muy interesante. Si os parece, pasamos al siguiente tema. Creo que bueno, se ha tocado, pero ¿qué opináis de irse fuera a hacer vuestras estancias, colaborar con gente, irse de postdoc? ¿Lo creéis necesario? ¿Lo creéis dependiendo del caso? ¿Creéis que ayuda al sistema científico? ¿Creéis que debería ser obligatorio? Bueno, empezamos otra vez por el principio, Nati, Jim.
Bueno, obligatorio, yo creo que pocas cosas en la vida deberían ser obligatorias, ¿no? O sea, creo que yo hablo a nivel personal, a mí me ha aportado muchísimo hacer la tesis fuera y hacer estancias fuera, tanto a nivel personal como profesional, pero obligatorio creo que cada persona tiene sus necesidades, tiene sus circunstancias y hoy en día es lo que se está pidiendo, casi una obligatoriedad para acceder a puestos y no me parece justo tampoco, o sea, te limita mucho a, dependiendo, tienes que haber seguido tu carrera en unos determinados años, estar dispuesto a moverte si ya tienes familia con tu familia, muchos condicionantes que quizá a nivel personal no te compensan. Entonces, hay muchos casos en los que bueno, que creo que se demuestra que aunque no te vayas fuera, tienes capacidad. O sea, otra cosa es que tú te quieras enriquecer a nivel personal y profesional, pero ¿por qué no?
Jim, que viene de fuera, es lo reverso.
No, es que en mi opinión, solo puede ser una cosa buena, porque puedes ver perspectivas diferentes de sistemas diferentes de ciencia en otros países que todos son distintos. Y también es una manera para crecer tu red de conexiones, porque la gente que, si es una estancia pequeña, bueno, Laura, tú sabes que estás allí 2 meses en Boston, vas a hacer colegas allí que vas a tener toda tu vida. Es una cosa muy breve, pero muy intenso, y tienes estas conexiones científicas que no puedes tener solo por si quedas en la misma instituto toda su vida científica. No es lo mismo, no es lo mismo. Esto es mi opinión. No tiene que ser obligatorio, que, bueno, depende. Lo sé, el sistema de financiación europeo está empujando este intercambio de países, pero es una cosa político de la fuente de financiación. Pero bueno, estas son cosas, bueno, opiniones diversas de mí.
Sí, yo estoy también con Natalia y Jim, que es al final irte fuera pues es muy enriquecedor, tanto desde un punto de vista científico como personal. En mi caso también me sirvió mucho para crecer como persona y he hecho amigos y he hecho interacciones que a día de hoy las mantengo. Pero creo que no debería penalizar. O sea, creo que no debería, porque a día de hoy yo creo que eso, que está, la carrera científica está enfocada a eso, a hacer la tesis, después hacer un postdoc fuera y después volver ya de vuelta o para con una posición más estable o lo que sea. Entonces creo que sí debería haber la opción también de que si tú no quieres ir fuera porque tú no te apetece, porque tu condición personal no te lo permite o lo que sea, pues que tampoco te vieras penalizado por no ir.
Rosa o Borja o Laura, ¿algo que añadir?
Bueno, yo estuve fuera muchos años. En las épocas que yo me iba era te vas y ya veremos cuándo volverás, ¿no? entonces ahí pasábamos años ahí fuera, un poco en la incertidumbre. Siempre fue positivo para mí estar fuera porque ves maneras de hacer las cosas que yo no había visto en los sitios en los que había estado, ¿no? En los laboratorios que había estado, sobre todo eso de una manera de enfocar la ciencia, etcétera, no ya aparte de los medios que pueden haber en otros laboratorios, etcétera, comparado con muchos laboratorios en España, pero bueno, ciertamente enriquecedor. Obligatorio no, por supuesto, porque cada uno tiene unas circunstancias personales diferentes y pueden ser muy diversas, ¿no? Pero si tienes la oportunidad, está bien. Bueno, pero aquí otra vez Laura Merino y Borja, que son los que lo tienen por delante, a ver qué dicen.
Bueno, yo en mi caso justo el mes que viene me voy de estancia, o sea que tengo esa suerte.
¿A dónde te vas, Borja?
A Austria.
¿Se puede saber? Muy bien.
Y entonces yo, bueno, es que yo creo que cuando te vas fuera también te das cuenta de cosas como técnicas como muy básicas que a lo mejor en tu laboratorio desde que entraste hacías de una forma, llegas ahí y resulta que lo hacen de otra y a lo mejor ni te lo habías planteado, ¿no? Ah, pues esto se puede hacer así. Entonces, yo creo que es como un plus muy, muy grande. O sea, no es lo que estamos diciendo, no digo que sea obligatorio, pero claro, si te dan muchísimos puntos para conseguir una beca, es como que se acaba convirtiendo casi en obligatorio, ¿no? Entonces ahí está el tema, cuánto potenciarlo. Pero yo sí creo que te potencia un montón. El problema es la gente que no puede irse, claro. Pero aprendes, yo creo que aprenderás muchísimo, vamos.
Bueno, yo todavía no he tenido la oportunidad de poder hacer ninguna estancia, pero bueno, un poco lo que habéis dicho, sí que tengo ganas y creo que tiene que ser algo enriquecedor tanto a nivel profesional como personal. Y luego también, bueno, a ver, dependiendo de la situación personal, pero creo que al menos cambiar de laboratorio en algún momento y ver diferentes formas de actuar o de cómo ver la ciencia siempre van a ser buenas para tú también pues, poder enriquecerte, aunque no sea fuera de España obligatoriamente, pero sí al menos eso, tener cierta diversidad de opiniones y de cómo ver la ciencia.
Muy bien, no tengo nada más que añadir, estoy de acuerdo con todos los puntos de vista. Si os parece, para ir terminando, vamos a dar alguna recomendación de un libro científico o relacionado con la ciencia que os haya gustado. ¿Tenéis algún libro que recomendéis para la audiencia que pueda ser interesante? El primero que se os ocurra.
Catálogo de Sigma.
Sin duda muy recomendable. Actualmente,
A mí no me parece muy interesante.
Durante mi tesis, eso ya fue el siglo pasado, claro, utilizábamos el catálogo de New England Biolabs. No estoy haciendo aquí publicidad de nada, porque era maravilloso para biología molecular. O sea que cuidado con los catálogos. Yo de literatura así más de divulgación o de dissemination, siempre todos los de Stephen Jay Gould, de evolución, maravillosos. Todos los de Oliver Sacks, a mí también me parecen todos maravillosos y muy informativos. Y últimamente uno que he leído más recientemente, que me ha gustado, Bill Bryson creo que se dice, el cuerpo ocupante, no sé cómo se dice en castellano, ¿cómo es? Guía para ocupantes. Se llama, que también está muy bien. Cualquier libro de este hombre, aunque no sea relacionado con la ciencia, es muy, muy interesante. O sea que sí que lo recomiendo.
¿Alguien más tiene una recomendación?
Yo no es muy científico, pero me ha gustado mucho y creo que ahora que estamos reivindicando también mucho el papel de la mujer en la ciencia, es muy bonito de leer. Es sobre Marie Curie, sobre su vida, La ridícula idea de no volver a verte, de Rosa Montero. Muy bonito. También recomiendo, ya no es libro, pero la película que la vi el otro día de Radioactive, me parece que cuenta, es la nueva de Marie Curie, es del 2020 o así, y cuenta la investigación en sí de una manera muy divulgativa y muy entendible para el público en general, y me gustó mucho.
Vale, apuntados quedan. Yo recomiendo un autor que descubrí no hace tanto, que se llama Siraja Mujer, quiero decir, es indio, no sé cómo se pronuncia, pero escribió para mí el libro científico de divulgación por excelencia, que es El emperador de todas las enfermedades, creo que se traduce en castellano, y posteriormente también escribió el de Gene, el gen, también súper recomendable. Y a mí me cambió la visión de ver el cáncer bastante, tengo que decirlo. Intenté leer Stephen Hawking, Historia del Tiempo, y lo dejé en la segunda página, así que ese no lo recomiendo personalmente. Aunque dicen que es muy interesante para los físicos, pero en mi caso particular no.
Y Laura, esta misma autor ha escrito durante la pandemia, ha escrito un libro nuevo, se llama El Canción de la Célula. Y es un resumen de toda la biología celular básico, pero también aplicada a enfermedades, y está súper bien. Entonces, muy interesante.
Perfecto. ¿Alguien más? ¿Algo que añadir a este aspecto? O ya paso a la última pregunta de la sesión de la mesa redonda. Vale, me gustaría hacer un juego con vosotros. Esto lo hemos ideado entre Rosa y yo, no soy solo culpable. Nos gustaría deciros dos, bueno, en realidad hay 4 palabras, depende de lo buenos que seáis, diremos 3 o 4, pero tenéis que venir, abrir el micrófono. Voy a dividir en 2 grupos, vamos a decir. Primero abre el micrófono Nati, después Borja, para que sea espontáneo. Y Laura, digo la palabra y vosotros decís la primera palabra que os venga a la cabeza con este concepto, ¿vale? Así tenemos 3 palabras, pero una viniendo de Nati, otra de Borja y otra de Laura. ¿Lo entendéis, verdad? Vale, pues tenéis que ser rápidos porque tiene que demostrar lo que...
Que nos acaba el tiempo,
que
así que
somos.
rápido.
Eso es. Vale, esta es facilita. CRISPR-Cas9.
Cloning.
Innovación.
Oh,
Cloning,
Innovación.
innovación.
James Sutherland.
The Master of Cloning, The Master of CRISPR. Vale, para Juanma y mi Rosa, os voy a poner una poco más peliaguda, ¿vale? ¿Jefe o jefa?
Jefa.
Duro.
Jefa.
Jefe o jefa, uf.
¿Podéis decir algo que os venga a la cabeza de jefe, de la figura del jefe, o podéis decir entre jefe y jefa? Bien. Jefe o jefa, rápido.
Jefa.
Buena persona.
Un trabajo duro.
No.
Vale. Y si queréis terminamos con una también un pelín peliaguda, pero podéis participar todos. Salarios en la ciencia.
Bajos.
Bajos.
becarios.
Becarios.
Deficientes.
No sé.
Unanimidad aquí.
Ya
Con
te digo.
tu cara lo dices todo, Jim.
Sí, bueno.
Yo diría irrisorios y muy mejorables. Así que a ver
Mejorables, vamos a poner.
sí, a ver si esto cambia en el futuro. Y ya la última porque me parece bastante interesante, esta la propuso Jim. Si tienen que hacer una película sobre vuestra vida, ¿qué actor principal quisierais que os interpretara?
Nosotros decidimos.
Sí.
Depende en qué época de su vida, ¿no?
De la vida.
Sí,
Podéis
de la-
elegir.
Uf, madre mía. Por supuesto, Angelina Jolie.
¿Alguien más?
Yo
Puede
Javier
ser,
Gutiérrez.
puede ser chico,
¿Qué?
aunque seáis chica y viceversa, porque os
También.
veis más identificado con la personalidad.
A mí me gusta mucho Marion Cotillard, ¿sabéis?
Ujú. Sí.
Pero porque me gusta ella, entonces digo, ojo, pues estaría guay, ¿no? Pero...
Yo lo mismo con Javier Gutiérrez, me gusta mucho como actor, entonces pues por eso mismo.
Bueno, por mi aspecto en la pantalla que veo así, Joaquín Phoenix.
O Brad Pitt, James.
Oh, gracias.
En mi caso, no sé, a mí porque me gusta mucho la peli así, pues Keanu Reeves, la peli de Matrix me encanta, entonces
Oh, Keanu, sí.
Muy bien.
encima tengo un look parecido.
Te pareces, te pareces. Sí.
Vale, ¿y tú, Laura?
¿Y Laura?
Yo no tengo ni idea. Yo qué sé. Zendaya, venga.
Bueno,
Vale,
puede ser Rosalía y es un musical.
Bueno, me parece bien también.
¿Y tú, Laura?
Difícil, me gusta como personalidad, me gusta Steve Carell, el que hace la serie de The Office, porque me río muchísimo con él y creo que tenemos muchas cosas en común en personalidad, obviamente es un hombre, pero me gusta mucho Jennifer Aniston, me parezco un montón a ella. Vale, pues chicos,
Bien.
ha sido un placer esta mesa redonda y también las presentaciones, agradecer de nuevo a la Sociedad Española la SEB, su invitación y es un placer siempre que cuenten con nosotros. Nada más que añadir.
Yo también eso, quiero dar las gracias a Sara y a María y al resto del equipo por el trabajazo que hacéis organizando estos Elevator Pitch, que creo que están muy, muy bien. Así que muchísimas gracias.
Nada, muchas gracias a vosotras. La verdad es que hacéis siempre un trabajazo seleccionando charlas súper interesantes. Se crea un debate muy rico y lo decimos siempre, la mesa redonda nunca deja de sorprender. Esta última parte, por ejemplo, no lo habíamos visto nunca. Esto de las recomendaciones literarias, los juegos, me ha gustado mucho la novedad, la innovación. Así que nada, muchísimas gracias a todos por participar. Ha sido otro elevator estupendo. Mucha suerte a los que estáis ahí todavía con vuestra investigación predoctoral. A los postdoctorales ya ni os cuento, que además es con quien más me identifico, así que... Y nada, a los senior que sigáis haciendo ese estupendo trabajo de mentores que hacéis con todos nosotros todos los días. Y siempre lo decimos, con lo que nos maltrata este país a los científicos, algo tiene que tener este trabajo que seguimos al pie del cañón.
Así es.
Así que
Así es.
muchas gracias a
Muy bien.
todos y a los
Gracias.
asistentes, por supuesto.
Gracias.
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 Adiós.